welcome to Biology, the scientific study of life. Biogeographic factors affect community biodiversity. Communities vary greatly in the number of species. They contain two key factors correlated with a community species diversity are its geographic location and its size. In the 1850s, both Charles Darwin and Alfred Wallace pointed out that plant and animal life was generally more abundant and varied in the tropics than in other parts of the globe. Darwin and Wallace also noted that small or remote islands have fewer species than large islands or those near continents. Such observations imply that biogeographic patterns in biodiversity conform to a set of basic principles rather than being historical coincidences. One way to understand such large-scale patterns in the diversity of life is to examine variation along environmental gradients. Equatorial polar gradients. As Darwin and Wallace noted, tropical habitats support many more species than do temperate and polar regions. For example, one study found that a 6.6 hectare, 1 hectare equal 10,000 meters square area in Sarawak, part of Malaysia, contains 711 tree species, while a 2 hectare plot of deciduous forest in Michigan contains just 10 to 15 tree species, and the whole of Western Europe, north of the Alps, an area of more than 2 million kilometers square, has only 15 tree species. Similarly, there are more than 200 species of ants in Brazil, 73 in Iowa, and only 7 in Alaska. The two key factors in equatorial polar gradients of species richness are probably evolutionary history and climate. Over the course of evolutionary time, species diversity may increase in a community as more speciation events occur, and tropical communities are generally older than temperate or polar communities. This age difference stems partly from the fact that the growing season in tropical forests is about 5 times longer than the growing season in the tundra communities of higher latitudes. In effect, biological time and hence time for speciation runs about 5 times faster in the tropics than near the poles. And many polar and temperate communities have repeatedly started over as a result of major disturbances in the form of glaciations. Climate is likely the primary cause of the latitudinal gradients in biodiversity. The two main climatic factors correlated with biodiversity are solar energy input and water availability. These factors can be considered together by measuring a community's rate of evapotranspiration, the evaporation of water from soil plus the transpiration of water from plants. Actual evapotranspiration, which is determined by the amount of solar radiation, temperature, and water availability, is much higher in hot areas with abundant rainfall than in areas with low temperatures or low precipitation. Potential evapotranspiration, a measure of energy availability but not water availability, is determined by the amount of solar radiation and temperature and is highest in regions of high solar radiation and temperature. This species richness of plants and animals correlates with measures of evapotranspiration, as shown in this figure. Area effects. In 1807, Alexander von Humboldt described one of the first patterns of biodiversity to be recognized, the species area curve, which quantifies what probably seem obvious, all other factors being equal. The larger the geographic area of a community, the greater the number of species. The likely explanation for these patterns is that that larger areas offer a greater diversity of habitat and microhabitats than smaller areas. In conservation biology, developing species area curves for the key taxa in a community helps ecologists to predict how the potential loss of a certain area of habitat is likely to affect the community's biodiversity. This figure notice that is a species area curve for North American breeding bears with breeding populations in the mapped area as opposite to migrant population. The slope indicates the extent to which species richness increases with community area. The slopes to different species area curves vary depending on the taxon being sampled and the type of community, but the basic concepts of diversity increasing with increasing area applies in a variety of situations, from surveys of ant diversity in New Guinea to the number of plant species on islands of different sizes. In fact, island biogeography provides some of the best example of species area curves. Island equilibrium model. Because of their isolation and limited size, islands provide excellent opportunities for studying the biogeographic factors that affect the species diversity of communities. By islands, we mean not only oceanic islands, but also habitat islands on land such as lakes, mountain peaks, separated by lowlands or natural woodland fragments surrounded by areas disrupted by humans. In other words, any patch surrounded by an environment not suitable for the island species. In the 1960s, American ecologist Robert MacArthur and E.O. Wilson developed a general model of island biogeography, identifying the key determinants of species diversity on an island with a given set of physical characteristics, as shown in this figure. Consider a newly formed oceanic island that receives colonizing species from a distant mainland. Two factors determine the number of species that will eventually inhabit the island: the rate at which new species immigrate to the island and the rate at which species become extinct to the island. And two physical features of the island affect immigration and extinction rates: its size and its distance from the mainland. Small islands generally have lower immigration rates because potential colonizers are less likely to reach a small island. For instance, birds blown out to sea by a storm are more likely to land by chance on a larger island than on a small one. Small islands also have higher extinction rates as they generally contain fewer resources and less diverse habitats for colonizing species to partition. Distance from the mainland is also important for two islands of equal size. In closer island generally has a higher immigration rate than one farther away. And because of their higher immigration rates, closer islands also have lower extinction rates, as arriving colonists help sustain the presence of a species on a near islands and prevent its extinction. At any given time, an island's immigration and extinction rates are also affected by the number of species already present. As the number of species on the island increases, the immigration rate of new species decreases, because any individual reaching the island is less likely to represent a species that is not already present. At the same time, as more species inhabit an island, extinction rates on the island increase because of the greater likelihood of competitive exclusion. These relationships make up MacArthur and Wilson's model of island biogeography, as shown in this figure. Immigration and extinction rates are plotted as a function of number of species present on the island. This mathematical model is called the island equilibrium model, because an equilibrium will eventually be reached where the rate of species immigration equals the rate of species extinction. The number of species at this equilibrium point is correlated with the island's size and distance from the mainland, like any ecological equilibrium. This species equilibrium is dynamic. Immigration and extinction continue, and the exact species composition may change over time. MacArthur and Wilson's studies of the diversity of plants and animals on many island chains, including the Galapagos Islands, support the prediction that species richness increases with island size, in keeping with species area theory. Species count, as shown in this diagram, inquiry. Species counts also fit the prediction that size or the number of species decreases with increasing remoteness of the island. In the past several decades, the island equilibrium model has come under considerable fire as an oversimplification its predictions of equilibria in the species composition of communities may apply in only a limited number of cases and over relatively short time periods where colonization is the main process affecting species composition. Over longer time periods, abiotic disturbances such as storms, adaptive evolutionary changes, and speciation generally alter the species composition and community structure on islands. However, more important than the model's widespread applicability is its impact in stimulating this discussion and research on the effects of habitat size on species diversity, a topic of vital importance for conservation biology.